So you got a garden, you have these weeds, and there are two types of weeds. One of the weeds, the dandelions, they sort of need, in some sense, less resources, so they outcompete this green weed, right? So now you say, okay, but you don't know that, right? You just look at all this field of dandelions and you say, I don't like, actually, you should leave them there. But a lot of people will do a little bit of chemical warfare, right? And what happens when you do your chemical warfare? At the end of the chemical warfare, you got rid of all these guys that have a competitive advantage in the absence of the chemotherapy, and what happens? These guys take over, right? So we got rid of the ones that we got rid, and in a niche-like fashion, the ones that did not get killed got stronger. So that's sort of the topic of what I want to talk about. A bunch of people here talked about treatment holidays and things of that sort. So there's a lot of work in this area, even clinical trials. The way I like to think of the different, these terms were bandied around earlier, so let me just, my view of how I think of these different kinds of therapies. You have the standard maximum tolerated dose, okay, where you give a dose, okay, and then you wait a long time until you give another dose, okay, and that might work or not, depending on your context. There's a different kind of therapy. Normally, many people call that metronomic therapy, right? And in metronomic therapy, the people like Bob Gadenby have very much worked on this kinds of, Gadenby worked more on adaptive, but it's not unrelated. In the context of metronomic therapy, you give lower doses, but you give them more often, okay? You can also have adaptive therapies. What's an adaptive therapy? An adaptive therapy is one in which you constantly monitor what's happening, and you will take an action when something bad happens. For example, the patient reports insomnia. I don't know, or no, that was a joke because that's another one. But maybe some marker is really bad, or there's a toxicity sign, and at that point you sort of don't do any therapy for a while, and then you do it again. So you wait for some triggering event, and then as a control freak that I am, control theorist that I am, you also have what you might call full feedback therapy. So those things are all what is called open loop or feed-forward therapies. But you can also have feedback therapies where you're constantly monitoring what's happening. And of course, you can't do that right now, but in the future with infusion pumps and liquid biopsies measuring circulated tumor cells and things of that sort, we can see that in the horizon as a possibility, right? So for now, it's just science fiction. If I would be Musk, I would say we're going to have that in 6 months, right? Full driving self-therapy. Anyway, we're being recorded. Can we get sued? Anyway, all right. So, okay. So as we heard many times already today, and we will during the rest of the workshop, resistance is a major contributor to cancer recurrence. This can happen due to many, many different pathways, many different things, okay, that can happen. Lots of pathways in there that contribute to resistance. But I want to focus on one specific thing, and the word Lamarckian was just brought up by Herbie. One of the questions that people ask classically is, suppose we have that there is some difference, pre-existing maybe, in your population. Some cells are more resistant than others, whatever that means. I don't want to get into the definition of what that means, epigenetic, genetic, and so on, but I mostly think of epigenetics. And then what might happen is that you have this mixed population, and now in a purely Darwinian selection, classical Darwinian sense, when we apply therapy, we're going to select for the resistant population, okay? Or maybe persistent, whatever, different kinds of resistance, stem cells, whatever. However, it could be that, and this is a classical question going back at least to Lurie and Delbruck, I'll remind you of that later in the next slide, it could be that the resistance pre-existed, or maybe the resistance arose during the treatment, okay? But what I want to emphasize in here, and that's the topic of the lecture, is that when the resistance arises in a treatment, it might arise due to a purely Darwinian selection, or it might arise in a Lamarckian sense, meaning that we also generate variations. We don't just select, but we generate variation, okay? So the giraffes are really stretching their heads to catch the things on the trees, and that gets somehow inherited, okay? So one question is, well, how can we differentiate between those? And that's what I want to talk about. But before that, let me remind you, just for the sake of completeness, we go back, 84 years or something like that, to the work of Lurie and Delbruck. And this is classical for 90% of the people here, but maybe some people are not so used to it. They were interested in this question: is resistance, does resistance arise during treatment, or does it pre-exist? And they did this very beautiful experiment, which leads to a lot of good probability theory when you want to do it right and everything, right? But the idea would be that you first culture in a non-selective medium and do many passages, whatever, and then you put things on a plate and you treat them with, in this case, a bacteriophage, which is a virus that infects bacteria, but could have been antibiotics or something else. But you try to kill, you try to see what's going to happen. And then if you have that the population is very homogeneous and only when you plated it, you get mutations, you will get basically a binomial distribution if things are happening at random. So basically you get a Poisson distribution of the number of the different, you try this on many different plates, and you get a binomial distribution. You get 1 or 2 or maybe 3 with some probability, something around the mean. But basically the variance is going to be the same as the mean. Okay, so you can try that. Now, on the other hand, if resistance arose was pre-existing before the time that you played it, then you're going to have some lineages in which, in some of these plates, you're going to have a number of cells which are already quite large to start with that are resistant. So when you actually plate, you're going to get a highly non-Poisson process. So at least that was their idea, and they did this work, and it was a beautiful, beautiful work that starts called the fluctuation experiment. And of course, again, most of you are very aware of all this. So again, this is what I already said. I would like to distinguish now not between pre-existing and resistance that happens during treatment, but I like to look at this finer distinction, which is not usually made, which is resistance that arises due to the treatment itself through Lamarckian followed by Darwinian selection or purely Darwinian, okay? Now, when I talk to biologists about this, they say it's the same thing, who cares, okay? So I want to sort of point out that I think one should care about it, because if you have first of all, how do you even define it? Okay, and it turns out the mathematics is trivial how to define the difference. You'll see in a second. I mean, I'm sure you already figured it out. But could it have consequences in terms of treatment? And this is all, I haven't done any treatment, but the idea. So resistance can be induced by drugs in many ways. We heard a lot of things already through many people, so I'm not going to go through, the various kinds of metabolism is there too, by the way. So as I'm saying, they're basically, I'm repeating myself. I'm really repeating the same thing over and over again. I'm saying we could have resistance that was pre-existing, or you have resistance that appeared during the application of the therapy. And that resistance could be spontaneously acquired, randomly happening in some sense, maybe reversibly, or it could be induced by the treatment itself. And of course, you could have combinations of things. So what got us started in this project already several years ago was this beautiful paper by Shui Huang's group, with Angela Pisco was the first author, in which they had a particular kind of, you're a mathematician, it doesn't matter, it's just histograms, right? That's all we care. But if you really care, the leukemia type cell, et cetera. And they were treating with a standard chemotherapy agent. In the second part of the talk, I'll talk about targeted therapy. But right now, just standard chemotherapy. And what they found is that they could actually measure the expression. They did a lot of single cell stuff, and they could measure the expression of MDR1, multi-drug resistant a gene, which expresses a certain pump that will basically pump the drug out of the cell. Okay, so that's the way they resist. They started generating these pumps that go to the cell membrane, and it says, I don't want this drug, send it out of the system. And what they find is that depending on the amount of these particular drug that they're vincristine that they're applying, these histograms of the amount of expression of this MDR1 gene, which think of it as a proxy for how resistant this population is. There's a histogram in that population. I'll get back to that in a second. And you see that the graph sort of shifts in a way that is, in some sense, non-linearly proportional to the drug amount. So what's really cool, and people were talking about Markov chains before, so let's revise that for a minute. I'm not going to talk about probability models, but that's the easiest way to understand what's going on in this case. This is an actual, the resolution is terrible, I'm sorry. So this is a cell number histogram of the amount of NDR expression. And what you have is that in the wild, when you're not applying any drug, you have an interesting distribution of resistance. In your population, you have, because of some random equilibration, think of a two-state Markov chain, A goes into B, B goes into A, there's some equilibrium probability distribution. It's a little more complicated. It's a continuum of resistance in the way that Herbie was talking. I don't have a PDE here, but it's sort of the same idea. So you have this continuum of resistance. And what's really cool about this is if you look at the cells that have MDR1 low, which means they don't have too many of these pumps, which means they're actually sensitive to the drug, okay? And you look at the ones that, let's say, the top 2% that have something like a very high expression of this resistance. You take out the cells using some flow cytometry, right? Cell sorting through flow cytometry. You take out these guys, and within 12 hours, you get back to the equilibrium probability distribution. I mean, if that's not a beautiful Markov chain equilibrating, you tell me one that is, okay? And if you actually take the guys that are highly resistant, after about 17 days, you get back the distribution. Yeah.
That means that it's not genetic, right?
Oh, no, of course not. It's all epigenetic. I mean, it's MDR1. It's expressed through, I'm not sure if people actually know the exact signaling mechanism. By the way, I do know it in some of the EGFR examples that one of you was talking about. There is more through, well, I don't want to get into that, but in the case of MEK inhibitors and stuff, one understands the feedbacks. There's a negative feedback loop that gets disrupted back through the SOS pathway to EGF receptors. But in this case, I don't know the exact, but yes.
It's not genetic.
It's not genetic.
When you say Lamarckian,
epigenetic.
When you say Lamarckian, that's genetic.
It's not, yeah. Well, Lamarck wasn't thinking of genetics and epigenetics.
12 hours is probably not even epigenetic.
Sorry?
12 hours is probably not even epigenetic.
Sorry. So I'm sorry. So we have to be careful with our definitions.
Yes, I know.
can give you a mathematical definition of epigenetics. One definition of epigenetics is histone modifications and the stuff that we do in our papers. Another definition people like, and they like it in particular, Shui Huang has written N papers about this. You think of epigenetics as simply having maybe two domains of attraction, and you are in one of, you push things like a bicycle.
Original Waddington definition.
Yeah, except that with reversibility. But yes, exactly. Okay. So, Pisco, yeah.
Question about how the experiment's actually done.
No, don't ask me. Well, don't ask me, but I know an answer, but okay.
So you take out the cells that have low expression.
Yes.
And you put them somewhere else where they continue to proliferate, or they continue to.
Yeah, they live like no drug. They live happy.
They just live happily, and then after 12 hours, you just look at them again to see the population of the percentage of cells that have the different.
Yeah, and you sequence them, and you find the one. Oh, actually, don't see. I think it's fluorescent marker.
So have the cells divided a lot of times in the 12 hours?
Well,
so
these are. It's a good question. These are mammalian cells, right? So it's for like 24-hour cell cycle. So
these are actually cells that haven't divided, but are actually just changing their metabolism.
Well, I don't know if it's metabolism.
Something like
some signaling, some gene expression, let's say. Some gene expression. I wouldn't call it metabolism. Yeah, it's an excellent question. I haven't actually thought about that, but this are. Again,
so somehow the other cells know about the other cells.
Sorry?
It would mean somehow that the cells in the plate know about the other cells in the plate.
No. You're flipping coins. You and I, we're flipping coins, and with 20% probability, we'll get one thing or the other.
Each individual cells.
Yeah. They did single cell analysis.
Doing some sort of random walk.
Oh yes, exactly, a random walk. Yes. So anyway, yeah, it is. No. It's think of it as a random walk, but even in a discrete ladder.
No, I understand. Yeah. I mean, it's like a random walk.
Yeah.
Some state space.
That's converging pretty fast. Yeah.
Sorry.
That's converging really fast.
It's converging fairly fast. I mean, I think this is fascinating. I mean, when I saw
So it means that the cells, that means that in a plate of cells, they're constantly changing state.
yeah, very likely. Yeah, that seems to be the case. I mean, everything is consistent with that. Yes, yes. Of course, you can. It's hard to measure the same cell, right? Because you're doing flow cytometry, and you know.
Oh,
so,
I understand. I understand.
but
I
it's
can't
in
imagine how you measure this at all.
population-wise. But yeah, yeah. Anyway, so I mean, this is basically answering both of the two questions. I mean, what both of you said. Okay. Which is tumor cells within a clonal population, the spontaneous switch between different metastable attractor states, okay? That's the definition of epigenetics in this context, okay? Which represent different environmental states, et cetera. By the way, you got your mesotelium, et cetera, in there. And then I'm just quoting from their paper, right? That you can think of this as Lamarckian induction, and Natasha doesn't like my using the term Lamarckian there, but I'm just using.
Does it mean something like that it increases the rate of variation, of mutation in effect?
Well, so that's
epigenetic mutation.
So let me get to that, because the point of what I want to talk about is sort of trying to understand if the drug is increasing the rate of some. In a sense, yes, it seems the drug is increasing because you can see about the survivability of the different. I mean, you can see how on this graph, right? You can see how the histogram is moving as you're applying different amounts of the drug, right? So you can see that it's sort of shifted, right, to the more resistant type. So when the drug was applied. So I want to say there's a number of people that have looked at drug-induced resistance, not just that paper, obviously, a whole bunch of people. Herbie was talking about that earlier too at the end. I'm only just flashing a bunch of papers there to point out that, these are papers, random papers from the literature that deal with this. So how can we differentiate? How can we differentiate between, again, I want to differentiate between resistance that is due to the application of the drug or resistance that appears spontaneously, okay, and that is being selected, right? The resistant cells are being selected because those are the ones that you can kill, and there's some competition between the resistant and the sensitive cells, okay, through some carrying capacity term in the logistic equation. So I'm going to give you a little bit of a mathematical formalism for this. I'm going to show you some simulations. I'm going to show you a little bit of theory. And then in the second part of the talk, shorter part, I'm going to tell you about some more recent papers that we have, which is this particular paper that just came out in MPJ Systems Biology. But those are the basic papers, okay? There's the ASCO paper. This one we actually never published. We didn't get around to publish it, still at bioRxiv. So what is a model? I couldn't be simpler, okay? So I'm all with René. I think simple models are good, okay? The simplest, the easiest it is to understand. I don't need a neural network with a billion weights to be able to understand things. So in this model, what we're going to do is we're going to have a logistic term so there's sensitive and there's resistant, and I'm just going to have one of each, okay? Towards the end of the talk, I'm going to talk about what happens if we increase the number of compartments, okay? To have more cells, and we'll see for some of this data, you don't get anything. So we're going to have a logistic term for the growth of S, logistics term for the growth of R. We're going to assume, that first. Second, I'm going to let V be the total, think of it, tumor size. So the number S plus R, typically that's the only thing you can measure unless you actually go and do some sequencing or you do some of this expression of this particular markers. So this is just a standard logistic, right? But notice that there's this competition term, right? Because V is the sum of the two, okay? We're also going to have this term, epsilon, which corresponds to a flux from notice the negative in there and notice the positive in here, okay? So this is epsilon times S, is some sort of flux that is occurring independently of the amount of the drug. U of T is going to be the amount of drug. I don't care about units. This is all math right now, okay? And we're going to have this parameter alpha that multiplies U. This parameter alpha is when you compare alpha with epsilon, this is the part that says how much the drug is producing the flux. So you got this valve, right? You have these two compartments, and you have this valve that you're turning, and the drug is turning that valve to have things flow from the sensitive to the resistant compartment. And I'm going to throw this out soon, but I'm going to assume there's reversibility, okay? And I'm also going to assume there's a death rate for both the sensitive and the resistant populations. And the obvious assumptions to make are that dr is less than d. I'm going to make dr equal 0 for simplicity, just for the mathematical analysis first. Okay, so dr, right, the resistant cells die less than the non-resistant cells. And I'm also going to assume that r sub r, the growth rate, is less for the resistant cells, corresponding to the fact that there's some, it costs metabolically or whatever to be able to make this resistance mechanism, okay? And these are all standard assumptions. So first of all, you could ask, is this parameter alpha identifiable? The answer is yes. You can do
Sorry, you also go back from resistant to susceptible.
Well, so yes, in this model, I'm allowing to have cells reverting from the resistance to the sensitive compartment. Most of the theory I'm going to show at the beginning is going to have that equal to 0. It makes the problem easier. But notice that there's still a connection between the two through the, that's very important, right? The competition term, right, is here. So the amount of R's that you have is going to influence your carrying capacity, okay? Now, that's really important because otherwise it would be trivial. Just have a cascade of two systems mathematically becomes trivial, yeah.
So do you ever consider, if you make gamma also have an alpha term, then that's like increasing the rate in both directions, but not favoring you one direction.
Yeah, a good question.
It's sort of the intermediate level of non-Darwinian, but not quite.
Yeah, I haven't. No, we haven't let the drug decide the rate at which you go back. You could, but in this mathematical, you have to choose your game to play.
That's fine. I just wanted to make sure.
Yeah, but it's excellent questions, of course. Okay, so theorem. I'm not going to give the proof of the theorem, a little thing, the usual things that you do in identifiability analysis. You can prove structural identifiability. So in principle, if you only measure the tumor volume, it turns out you have identifiability of alpha. So in principle, you could identify. But let me now get into what I think is far more interesting. So we talked a little bit before about the RECIST criteria. We talked about time to progression, right? So we're going to be very realistic in that way. So what we're going to do is we're going to try to understand how different therapies affect the time to progression, okay? So we would like to maximize the amount of time until the tumor became a certain size. Now, that's sort of interesting because when you do that, you actually have an optimal control problem, which is not the standard optimal control problem where you try to minimize the time for something, but you're actually trying to maximize, okay? So what we're going to do is we're going to assume that we start at some volume V0, and there's going to be a detectability volume, a detecting volume, and we assume that the therapies start when we get to the detecting level, okay? All right? And we're going to assume that we lost the game when we get to this particular critical value, okay? So there's going to be a critical value at which, if we get to the critical value, the game is over, and we're trying to maximize the time from the beginning until we get to this. Okay, sorry, you were talking about the RECIST criterion. Okay, So one question that, sort of an interesting question that I think the first question that we asked was, can we distinguish, because I said, most biologists, most oncologists would say, who cares? If you're going to have resistance, who cares if it was induced, it wasn't induced, okay? So what we want to say is that, as a matter of fact, this might have some implications in therapy. And in order to do that, the first thing I'm going to do is going to say, let's compare a continuous therapy, where you just apply a constant dose, to a dose in which I do, say, a metronomic type thing, a pulse therapy. So this is my inputs. It's a piecewise constant function, okay, with some periods of holiday in between, all right? And then, can we say something about how the parameter alpha, this induction parameter, the one that decides that things are sort of, quote, Lamarckian, even if Natasha doesn't like the word Lamarckian, that it's induced by the treatment, let me put it that way so I don't get in trouble. can that affect, can that tell us what happens? And the answer is, at least with a wide parameter sweep, and also some theory that partially, and by the way, in terms of open problems, I think really trying to understand this in a deeper mathematical way that we've done so far would be great, okay? Because we've done some parameter sweeps, we have a little bit of an optimal control result I'll mention, but the big picture, I'm not sure that we still understand. But in any case, the bottom line is that when alpha is non-zero, then don't worry about the units. It just seemed to make sense for us in terms of how the experimental data. In fact, the pulsed therapy is better in the sense that the time to progression is much longer. So the time to progression for constant is roughly here, okay, 42 whatever days or whatever it is. But if we use pulse therapy, we actually can increase the time to progression a fair amount. And in the case of of no alpha, where just epsilon is there, okay, where we don't have this faucet that has been turned by the water, but there's still this competition, remember, through the carrying capacity. In that case, actually, we get a far better, look at that, the constant guy, the pulse guy has this time to progression that is very little. The constant guy is much longer, right? So what it's saying is that the consequences of whether you do have alpha equal to 0 or not seem to be very important from the point of view of potential therapies, okay? Not that I'm going to convince anybody to do a clinical trial about this right now, but it would be great to do it with animals, and I haven't gone on anybody to do that, even in a good cell culture thing. All right, so let's consider a little more theoretical thing. What about if we think of the optimal control problem? So I'm going to tell you in a second, there's a constrained optimal control problem. The theory is quite technical. I don't want to go through all the details. I'll just give you the outline. But basically, the conclusion is that when there is no induction by the drug, then the optimal trajectory is simply a bang-bang trajectory in the sense of control theory with a boundary slide, which I'll explain what that means in a second. There are no singular trajectories in the interior of the state space. I'll explain that in a minute. And probably the most interesting thing about that is that when alpha is different from 0, and if alpha increases, then singular extremos can arise. However, the Legendre-Klebsch conditions from calculus of variation tells you they cannot be optimal. And in fact, you can actually do a very interesting little analysis using differential forms that I'll tell you in a minute. I'd like to sort of quote why using optimal control. Bob Gattenby, who's probably the most important person in this kinds of research, he said the oncologist has the advantage over the tumor of seeing the future and strategizing, including doing your computer experiments and projecting how things are going to happen. I love that phrase. Okay, this is just a little example from doing a numerical optimal control package for the problem. Did I have the actual statement of the, well, I had it in here, but I think I didn't. Yeah, okay, so it was it might be on my extra slides. I have to look in a minute. Okay, I have 100 extra slides in case somebody wants to ask more questions. So, here's a little example, okay, of what an optimal control looks like in this case. We have basically, if you look at the control that gets applied, the control gets applied is nothing for a while. Then you go into this particular sliding trajectory that I'll tell you about, and then you do a hit of maximum therapy. No therapy followed by, for this particular numbers, followed by this particular control. So, and there's a general structure that is of the form use either a full, which we didn't use. Oh, I'm sorry. I forgot. There is a full thing at the beginning. I'm sorry. I was trying to find where it was, and I couldn't see it on my slides. So you do full at the beginning, then you do nothing for a while, then you do this funny thing, and then you do full at the end. And that corresponds to the structure YXUPY, where Y is the vector field corresponding to U equals a full value. I'm assuming U is bounded between some values, capital 0 and capital M. X is a vector field corresponding to 0 value. So full value, 0 value. UP is this control that I'll tell you about. That is a sliding control. It's a feedback control. And Y is, again, just hit the thing with full force, okay? So, and by the way, of course, then you can see that what's really happening in there is the sensitive populations at the beginning grow a little bit when you are not doing anything here, but then they go down, but then the resistant guys are taking over. Okay, remember, we're only measuring the sum of the two, but the numeric in the control algorithm, in the analysis in control theory, you have the two variables. Do you have a question, Renee?
Yeah, I don't know if I'm missing it. So you're saying you're hitting them at the full dose?
At the beginning and at the end.
So,
It's not metronomic or anything. It's just the optimal control solution.
but they're both populations are starting at 0.
No, you don't see it there, but there's, I forget what you're starting slightly bigger than 0. Of course, if you start at 0, nothing's going to happen, right? Yeah, you can see it in there, but they're not starting exactly at 0. It's, I forget what it was, 0.01 or something. Yeah, we have less resistant than sensitive. I think we started with 0 resistance and a small sensitive. Yeah, sorry, I didn't have the numbers there. So anyway, so the theory results say something like the optimal controls will look in this form, so it's not quite bang, right? It would just be Y and X that we just bang, but you also have this particular type of control that I mentioned before. I'll show you a picture in a minute what that means. And we don't have this as a theorem yet, but the calculations seem to suggest that the number of switches that you'll need will actually go up when the induction coefficient is bigger. So, but we can't quite, prove that yet. Just to give an idea of the flavor, I'm not going to go through the details. Again, I have this in the extra slides, but you do the usual Lie algebra analysis that you do in optimal control theory. It works in any dimension, but you do in dimension 2, it's very explicit. You look at the switching function. We know the switching on singular trajectories, non-bang trajectories because of the maximum principle, Cherenkov maximum principle. We know that we need to minimize the Hamiltonian. We do that. In general, that's hard to do, but you take higher order derivatives to find the function that is multiplying the control. Eventually, that comes out through some, you take Lie derivatives of the corresponding function, right, with respect to the two vector fields, x and y, and then you end up writing out the form of the control, which end up being some complicated formula, okay, but basically you want to show that, I had the picture of so basically you have this line here that corresponds to where the switching, which potentially could be a singular trajectory. But what you do is you introduce you introduce this one form, so-called the clock form, that vanishes on the control vector field, but it's 1 on the drift vector field, and they're linearly independent. So you can always find such a form. You can actually compute it explicitly. Okay, as a one form, and then you can actually, it's a little calculation that it's quite standard, that the integral of this one form actually, the path integral actually gives you the difference in time from the beginning and the end. And to make the story short, you actually prove that doing something like I'm showing in there, it actually takes more time than going this way, which means that you never really stay along here because you want to maximize the time. Okay, so a lot of details about these things, and a lot of little undergraduate differential geometry or whatever, or analysis. Now we said, well, does this fit data at all? It turns out that model actually fits the data quite well. We censored a couple of times in which the data was obviously not right. But we actually, with this very simple model where it fit, very few parameters, where we could do a pretty good fit to the sensitive and resistant populations. And from just measuring the total. And then we extrapolated, so what would happen if there's no drug? If there's no drug, actually, you also got some pretty good fits. So it was sort of nicely validated on that. I don't have time because I want to tell you something else, but there was what I thought a very remarkable prediction about that repopulation experiment that I showed you at the beginning. You actually get, without doing anything and no probability or anything, you actually get, you didn't quite get the same as experimental data, but it wasn't too bad. Before I get to the next thing, just a very quick thing. There was a follow-up to this paper done with Amy Brock's group. Caitlin was her student at that time, PhD student at the time. And in this experiment, what was done was to take exactly what we had, exactly the differential equations that we had, to try to measure some tumor data. But in addition to that, there were 3 time points at which RNA-seq was done to find the expression of how much heterogeneity there was in the population. Anyway, when you the only, and I won't talk about this paper, it's this physical biology paper, but the point is when you put the two together, you could get some predictions that were actually much better than when you only fit the tumor data. So the bottom line from that is if you want to study this thing, somebody asked before about doing sequencing, certainly if you do RNA-seq, even with a limited number of points, you can improve your estimates tremendously in the model. So that was all good, but it was a little bit very simple. And we got a new set of data, and we started working with the first author of this particular paper, where this is a different problem. This is the one, but asking the same kinds of questions. This is a problem in which we have a targeted inhibitor. So this is not classical chemotherapy. This is a targeted inhibitor. A melanoma cell line. And I'm just going to tell you a little bit about the fits in this case. So what they did is they actually tested various, so one of the things that we want to understand is how does this induction happen, right? I mean, and it should somehow depend on the amount of drug that you're giving, right? The alpha multiplied U. Well, it turns out that model didn't quite work for fitting this data. I'll tell you what worked. But there were 5 concentrations in log scale, in 10 log scale, they were multiplied by 10. 5 concentrations of which we took 3 just to show you a fit, and then I'm going to show you how you extrapolate to the ones that we didn't use for fitting. And what's interesting in this data, this is the normalized cell count, normalized by the beginning over time. And at the beginning, all of them gave about the same. So that said, the model that we had was no good. We needed to have a delayed term of some sort. So we introduced a delayed term. Could be because metabolism of the drug, whatever it is. This is an in vitro experiment, so it's not PK/PD stuff, but not PK, right? But there's something happening with the drug. So we had I'm going to skip there's a 5-dimensional model, then you do a time scale decomposition. You end up with a model like this, okay? I don't expect that you're going to read the details of this at this time, but it's basically the same model as before, except that we have a little delay for the time that the drug takes effect, okay? And then we did some parameter identification, so identifiability, and we actually fit. The only thing, the reason I'm bringing this up is because I thought this was cool. We actually, there's a lot of the data. There's variances on the actual observations, right? And many cells, but the point is that we actually managed to get a fit. The red is our fit, okay? And we managed to get a fit to this data with this very simple model, okay?
So they have data like many times an hour?
Well, they're different cells. So there's a variance in here, right? So there are many
I'm
different
amazed
cells.
how detailed your data are.
Yeah. I think there were 4 replicates. Okay, it's actually written in here. So there are 4 replicates. So this is just showing it's hard to see. Yes, it's very time resolved, extremely time resolved.
Time resolution is amazing.
Super time resolved. That's why this was so cool data. Yeah, I mean, this is the data, right? Okay, for the means. I mean, but there were 4 replicates. So here I'm showing you it's noisy, right? I mean, these are non-trivial non-trivial variances. Okay, standard deviations, but still, it's pretty good. So what we did is we said, okay, we fit these parameters. Let's try to. Sorry, I'll get to that in a minute. But I was asking a question before. I think it was from Renee about the practical identifiability. And what was interesting was, okay, you can do identifiability using your algebraic geometry, or computer algebra systems and differential algebra, et cetera, et cetera. You can do all that. But also, you can just simply numerically try to find parameters that fit and do histograms of what parameters fit within, say, 5% of the optimum, okay? You have some cost function. You say, if this set of parameters is within 5% of the optimum, I'm going to throw it into this bucket. And then what you get is, notice that here there's these bars because these things are actually very separated. So they're actually very nicely separated for each of the parameters will be different for each of the dosages, okay? For each of the dosages, different parameters. But if we just pick one of them, for example, you can see these are the different parameters that were fit. And you see that you get very tight distributions, okay? This is important for what I'm going to say next, okay? So hold your thoughts for a second. But not this next, but next after this. So we actually said, okay, there were some dosages for which we had not used anything to fit. And actually, you get pretty good fits on those too. Not great. I still don't understand why this happens in there. There's a little bit of there should be more of a delay, but I didn't want to hardwire the delay and cheating. So you may ask, and this is what I was going to say about the histograms. You may ask, what about if we include quiescent cells? Now, in their experiments in this paper, they actually measure. They do a single cell analysis, and they actually see that there's some cells that are quiescent. Okay. So what we did is we said, okay, let's have a model with quiescent cells, everything the same. Let's fit this model. And what was interesting is we did not get better fits. The fits look about the same, okay? This is about the errors. Get the same errors when we fit with the three-compartment model. Remember, we only measured the total volume.
Did you run structural identifiability for this as well?
I'll tell you later. It's in the paper, but I don't. There's a small print thing. So let me answer. Yeah, yes, it's identifiable, but I hardwire gamma 1 and gamma 2 for reasons that I can explain later. Otherwise, it's not identifiable. But once you hardwire gamma 1 and gamma 2, the rest is identifiable, and the proof is in the paper. But what was interesting is that you get a huge dispersion in the parameters in this case. So it means that, sorry, that was for the first model I told you about. For the second model, it's not identifiable. That's the whole point. I'm sorry, I missed your question. You're asking me the question about the two-compartment model, the three-compartment model.
No, the new one you just
Yeah, no, it's not identifiable. It's not identifiable. Sorry, I was answering the question about the two-compartment model. It was identifiable. This one is not identifiable, but actually you can see it. You can also see it in terms of the dispersion on the parameters. They're all over the place. Okay, and this is doing single parameters, not doing double parameters or anything like that. But even on single parameters, you can see. And you can do an optimal control problem here too. I'm not going through the details. So I would say as a summary. I actually going to finish the talk in time. That would be the first time in my life. Anyway, all right. So
you're already 5 minutes.
Well, but that includes the question. So I mean, including the question period at the break. At the break. Anyway, so I told you, why. Why it is not necessarily a good idea to kill all the ones that you can kill. Okay, because you have the bad guys and you have the really bad guys. Okay, now if you do a no-fly zone and keep this bad guy under control, this bad guy is going to outcompete the really bad guys. Okay, now you go in and you invade and you get rid of this bad guy, and then the really bad guys can take over, and it's much harder to deal with them. Sorry, I didn't mean to be political or anything, but we're talking about weeds. Okay, all right. So sometimes it's better to keep the bad guys as long as we can control them, keep them, through some metronomic therapy or something, keep them to within a certain level so they can outcompete the really, really bad guys that is much harder to defeat. So that's sort of the big story, but I was concentrating on this question of how do we, is the variation induced or is it spontaneous? In other words, do we have a purely Darwinian phenomenon or do we have, a Lamarckian type phenomenon? Blame Shuey one. Anyway, and then, so what? Okay, so you write a little mathematical model and you can actually show for this mathematical model that one kind of therapy, pulse therapy, will be better if you have induced, and constant therapy is better if you have non-induced, and that might guide in the future our ways of thinking. So the way I look at all this work is not that you can take a picture of that and get it to where the papers are. Our collaborator, right, from the one paper, but I also want to mention Samantha, who was an undergraduate in my lab who did a lot of the work. Okay, so sort of nice to show undergraduates in there, like you did earlier. So the way I see this thing, and then I have a million other slides if anybody wants to see for another lecture. But what I really, my opinion is that, well, I don't know what my opinion is, but if I had to, since something about open problems and mathematics and all that, right? So I think that, I'm going to get in trouble if I say this. So I think cool mathematics is not the same as things that are important in biology, okay? So we can do as much algebraic geometry and whatever we want. I knew I was going to get in trouble. But that doesn't mean that we're going to do anything to cure cancer. We're going to have fun doing cool math. Now, one thing we can do is we can say, let's really talk to the biologists and understand what their problem is and try to abstract the problem. And then maybe understand that problem mathematically to get some ideas of what are the principles that should guide us. Okay, what clinical, even what preclinical experiments they can do, right? So guide sort of what the experimenters should do. But it's not like, from the community I come from, everybody's used to doing things to tolerances of 10 to the minus 7 because this airplane better has the right things and all that. But you can't do that in most biology, okay? So we have to learn to live with uncertainty. We have to learn to live with mixing maybe mechanistic and data-driven models. This is another paper just accepted on this, but a lot of people are working on that, but I'm saying I'm very interested in that topic. So I see mathematics as maybe, and for instance, you solve the optimal control problem, and of course the numerics of the optimal control problems are BS. I mean, nobody's gonna, those parameters are, made up and you're never gonna measure them right? But at least we'll give you an idea. Well, maybe we wanna look at a therapy which is a continuous therapy. And that actually can do better than any of these therapies that are used, okay? So maybe you should go and investigate continuous therapies. It doesn't mean I'm gonna get a 32% improvement or something, right? I don't believe any of those, okay? Of those, quantitatively, I don't believe any of the models, okay? But I think it's, sorry, I'm going into preaching mode. Okay, yeah, when I answer, maybe I should answer questions instead of preaching. Yeah.
For the continuous therapy,
yeah.
U is a function of the measured value of R plus S?
The U is a, yes, so it is. I wasn't actually talking, I didn't want to, so this is the other paper on the immune system. Which was interesting, but I want to get to, just want to get to the optimal control thing. So you can prove theorems, a bunch of interesting theorems, but you look at all the, measurable essentially bounded functions, blah, blah, blah. You try to maximize the time that you get to the B plus C equals V critical, right? So when you start around there and you want to maximize the time to get there, okay? And the U actually in there corresponded, I think I have in the next slide, sliding along this guy as long as you can do it, okay? So yes, you could think of it as a function of those, and I think the function was explicitly given there. I think I had it, yeah, there's a whole bunch of theorems about how you actually get it, but you get this explicit form, okay? I probably had it in one of the other slides, maybe not. But this is the whole derivation of the...
Which is the... Okay, I was trying to see.
Yeah, go ahead.
It looked like it required you to measure S and R separately to actually know what...
No, okay, good. Okay, excellent question, yeah. But before that, just a little point. So the analysis I was doing with the differential form and all that was to show that here, which is where you have singular trajectories, okay, that's a switching function that could vanish identically, those would not be optimal trajectories, okay, you would actually get out, and that was that analysis I did. This is a critical thing, and well, you don't need, yes, it's a very good point. If you were going to actually apply this, you would need to know S and R separately. Here, it was just to show that what the optimal solution looks like. You would have to, I actually hadn't thought about it because I was never talking about using this for therapy. I was just trying to understand the shape of the optimal solution. But you're absolutely right. It's not just a function of VC, it's a function of the proportions of the two. No, it's an excellent point. Yeah. If you, look, these are toys. I mean, nobody's gonna apply this.
Thank you, Eduardo.
I think Dominic had a question, but.